This working? Sounds like it. I hear reverb. Okay, good. So we finished up last time, and I asked you to just confirm something. We were looking at this equation that we derived for evolution at one locus with two alleles with selection, and we were at the time leaving mutation out. And we had an equation that looked like this. Change in allele frequency. You recall P is the frequency of A1. That change in frequency came out to look like this. There was a P times 1 minus P times the marginal fitness of A1 minus the marginal fitness of A2 divided by mean population fitness. The form of this equation, if you remember, we got this from the price equation. And we got it from a case where we wrote the price equation in terms of a regression of fitness on phenotype. And so, just to remind you, we had a term that looked like this. Delta phi bar was equal to a regression of fitness on phenotype times variance in phenotype. And then that could be, we could add a delta bar for changes occurring in the process of reproduction, but we were leaving that out, okay? This is a format that's going to come up all over the place. Basically, whenever you're talking about selection, any selection process, you're going to have variance of the thing selection's acting on times something that's going to be like a regression of fitness on that thing. And when you expand that and solve that regression for just about anything that you're interested in, you're going to find a 1 over W bar in it. 1 over W bar will keep showing up. Now, for the moment, this is just basically a scaling factor, because W bar is mean population fitness. It is the average across every individual in the population. So it doesn't capture differences between individuals. And it's often treated basically as something that you can almost assume away. One thing about, you can see from this equation, I could multiply my genotypic fitness values, the ones that go into calculating these, I can multiply those by any constant I want. The same constant will multiply every term in W bar. And so the constant will cancel out. So I can always scale fitness so that W bar equals 1, right? I'm going to leave W bar in here in our equations. And the reason that I'm doing that is that we'll see this is a deterministic model. When we make our description stochastic, then these W's are random variables, and W bar is a random variable too. And when you're dividing by a random variable, strange things can happen. So we'll want to keep track of it when we do that. But for the moment, we're working with this. And I set up a case. We had 3 genotypes, A1, 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 A2, and A2, A2. And I assigned fitnesses to these, W11, W12, W22. And from those, we got these marginal fitnesses. And they were W1 star was P W11 plus 1 minus P W12. And W2 star was P W12 plus 1 minus P W22. This is something y'all probably easily see through this, but when teaching this to students, especially when they're first coming to the subject, this is something that catches people up because they expect, well, this is an average overall genotypes that allele, the A1 allele for W1, appears in. Well, the frequencies of genotypes all have p squared terms in them, right? So why aren't there any p squared terms in here? These are essentially conditional probabilities. So this, for here, this is the marginal fitness of the A1 allele. It's saying if you're A1, what's the probability that you're paired with another A1? that's the probability that you're an A1 genotype, right? So we know one of them is A1, so we're asking what's the probability the other one is as well. That's just P. If you're A1, what's the probability the other one is A2? Well, that's 1 minus P, right? So if that's not clear, think about it. You can come up and ask later, but that's a distinction you want to be able to make. Now note, in doing this, we are assuming random mating, or most properly, we're assuming random union of gametes. Random union of gametes. That doesn't always have to happen. It's essentially always called in textbooks, it's called simply random mating. It is possible to have non-random union of gametes even if there's random mating. How could you do that? If individuals mate at random, but gametes don't actually come together and form zygotes at random.
If you have like some distortion in the meiotic process?
Yeah, so that will do it. If there's any sort of meiotic drive or segregation distortion, that's where meiosis itself gets mixed up, gets modified. Yes, that will do it. Another thing that will do it, think about it. So imagine individuals mate at random. Do all pairs produce exactly the same number of offspring? No, there's variation in that, right? And there's random variation, but there may be deterministic variation in that as well. Okay, it might be that certain genotypes for mom and certain genotypes for dad produce a lot more fertilized eggs than other combinations. That essentially would produce non-random union of gametes, even if individuals paired up at random. Take an extreme case, say they pair up at random, but whenever you have an A1 female and an A2 male, no zygotes can form. Okay? In that case, it's as though those gametes never come together because they never form any zygotes at all. So I'm using the more proper term here, random union of gametes, which is what goes into this assumption. Okay? If this were not the case, then it might be more or less probable if you're an A1 that you're paired with another A1. And you can think about what effects that would have. But anyway, so I set up and said, let's make this assumption. And then we wrote out mean population fitness. W bar, which is important, shows up here. And that was P squared W11 plus 2P 1 minus P W12 plus 1 minus P squared W22. These are just the Hardy-Weinberg frequencies. So we're assuming Hardy-Weinberg frequencies at the zygote stage at this point. And then I asked you to just confirm that the derivative of W bar with respect to P came out to be 2 times the quantity W1 star minus W2 star, which shows up here. Anybody check that? Did it work out? Anybody try it and it didn't come out to that? Okay, I'm going to assume that I'm going to go on. If you haven't checked that, I recommend you do it. If nothing else, that will solidify in your mind what's going on here, right? Because then you'll have to write this out and okay, take the derivative of it. And it's going to be a derivative with a bunch of W11s, W12s, and W22s in it. But you want to get it in terms of these W1 and W2 stars, and so you'll have to think about, okay, what do those mean in putting it together? So if you haven't done it, sit down and do it just for your own benefit, and then it'll stick in your mind forever. In fact, this is the case, right? What it means is this W1 star minus W2 star, that's equal to 1/2 the derivative of W bar with respect to P. And this yields from that an equation originally derived by Sewell-Wright back in 1932. You can write change in allele frequency as P times 1 minus P over 2 W bar, that 2 comes from dividing that, times derivative of W bar with respect to P. This is a very influential equation. Now, this is very simplistic in some ways. We're not only assuming random mating, we're also assuming frequency-independent selection. And I'll write that up again because that's really critical to this. Frequency-independent selection. And as I said, what that means is these Wij terms are not functions of P. So we're treating those as constants, essentially. If W11 was itself a function of P, then that would change the derivative of W bar with respect to P. So we're assuming frequency-independent selection. What does that mean? Pardon me. I'm inhaling roughly a third of a chalk piece here. What does this mean? This means, like I say, formally, these W's are not functions of P. They're multiplied by functions of P, but they're not functions of P. Biologically, what does it mean? It means that the fitness of, say, an A1A1 individual is not a function of how many A1A1s or A1A2s or A2A2s are in the population. So the fitness of an individual using a particular strategy is not a function of the frequency of those strategies in the population. Now, there are all sorts of cases, especially physiological traits, where that seems reasonable that that would be true. One example, I was talking to a couple of people asked yesterday about it. We're all fairly close to sea level here. We're a couple thousand feet up, but not particularly high up. If we all went up to 15,000 feet, we'd all, or many of us, would suddenly find it hard to breathe, right? We could breathe up there, but it's a lot harder to get a lot of oxygen just because the oxygen level's lower. When people move up to high altitudes, there's selection to either be able to bind, or there's selection to bind more oxygen in your blood, basically. And you can do that either by increasing the binding efficiency of your hemoglobin or by getting more hemoglobin. Okay, both of those things happen in different populations of humans who have moved to high elevation. But the point is, there's selection to be able to bind oxygen better in your blood, and that's really not a function of what anyone else is doing, right? That's just about you in that environment. Whether other individuals can bind a lot of oxygen or not doesn't affect your need to be able to do that. By contrast, a lot of behavioral traits, especially a lot of social behavior traits, are frequency dependent, not frequency independent. Then what you should do is a function of what everyone else is doing, right? So for a lot of behavioral traits, a lot of social interactions, whether you should do something or not is a function of what others are doing. And we mentioned yesterday, just very briefly, Fisher's sex ratio theorem. And that was an example where, to describe it, you look to the number of grandchildren, not to the number of offspring, right? So where you look over a couple of generations. The basic idea there is that if you have a skewed sex ratio in the population, if there are a lot more males than there are females, then there's selection to produce females. And if there's a lot more females than there are males, there's selection to produce males. And that pushes you back towards a 1-to-1 sex ratio. Even if the breeding sex ratio isn't 1-to-1, even if most males never breed and a few breed a lot, which is common in a lot of organisms, you still get pushed back to a primary sex ratio that's very close to 1-to-1. And the reason is, if the strategy is the sex ratio of your offspring, how selection acts on that depends on the entire population, right? So if everyone else is producing daughters, you produce sons. If everybody else is producing sons, you produce daughters. That's frequency-dependent selection, right? What you should do is a function of the frequency of what everybody is doing. We're not talking about that here. We're talking about frequency independence. So with that in mind, that we're assuming that here, and this is critical to interpreting this, This equation gives sort of a useful conceptual description, and Sewell Wright, when he derived it in a different way, he was very influenced by it, because here's a key thing. P times 1 minus P, P varies from 0 to 1, right? This is non-negative. It could be 0, it could be positive, but it's never negative. W bar is non-negative, okay? So this term here, this derivative of W bar with respect to P, determines the direction of evolution, because this could be positive or negative. And so whether the allele A1 increases or decreases in frequency depends on this term. Furthermore, this looks like gradient dynamics, right? It looks like the population's following a gradient. It is not exactly, because there's this scaling term in front here, okay? But especially when we're just talking about one locus, in terms of the direction of evolution, this looks a lot like gradient dynamics. And so what does that mean? Well, it means that in principle, and in fact, I can plot mean population fitness as a function of allele frequency. And if I get some function, say, that looks like that, then the direction of evolution is determined by the slope of that function. Over here, if I pick a point down here, the slope's positive, right? So the derivative is positive. And so since that's positive, delta P is positive, P increases. If I go over here, the derivative is negative. Since that's negative, the whole thing is negative because it determines the sign, and so delta P is negative and it decreases. And of course, when that's 0, the whole thing is 0. And so you can immediately see what sort of dynamic you have here. In this case, there's a stable internal equilibrium. And you can think of the population as basically climbing up this landscape. And this is one of the things that made people like to talk about the idea of an adaptive landscape. This notion that you can define a function, W bar, that is locally maximized. Do you have a question?
Oh, could you expand on what you mean by gradient dynamics? Is it something like a gradient descent algorithm, like we would think of?
So broadly, when I was using the term gradient dynamics, I mean a case where the strict gradient dynamics, the rate of change and the direction of the system is determined by the gradient of some function. And so in this case, and this is not really strict gradient dynamics, because the direction is determined by the gradient, right? The direction is determined by the slope of this function. The rate is not completely determined by that, because the rate's also affected by this term here. But what I mean is, in this case, yes, the direction you go in this case is determined by the gradient of some function. In physical applications, you often see gradient dynamics. Often it's flipped around. So the system approaches a minimum of some function rather than a maximum. But that's also gradient dynamics. It's just determined by the gradient, right? In this case, you climb uphill because of the way we wrote this. Does that make sense?
Yes. Just like you have an electric potential and then
Yes.
you consider something that's purely acting on the photon.
Yeah, exactly. So something that goes to a minimum of some potential function. Yes?
Could be the case here where we are bouncing back between two values instead of going to an equilibrium?
In this case, it's not visually obvious from this equation. I'll show you an equation later where you do bounce back and forth, and then I'll show you why this, with frequency-independent selection, this always actually does have local equilibria. Basically, losing parts of my chalk, if I plot p at time t, p at time t plus 1, 0 to 1, 0 to 1, what you get in the case for the frequency independent case, kind of looks like that. And as you make it stronger and stronger, if I make this peak really high in the middle, I approach that. It never goes past. So this slope never becomes negative for this equation. With frequency dependence, I can make anything happen. And I'll show an example later where with frequency dependence that becomes negative. Then you can make this an unstable equilibrium. But for this case, it's always a stable equilibrium. That's a good question, and because we will see in a little bit a case where it's not stable. Does this make sense, the basic idea that you're climbing up this landscape? Okay. And what this does is it gives us sort of an intuitive, or it gives us a formal notion, I should say, of the intuitive idea that a lot of people have about adaptation. That selection causes populations to evolve to become better at something, right? Remember, W bar is per capita growth rate of the population, right? It's the average contribution to the next generation. So I could write n of t plus 1 equals W bar n of t, okay? So what's being locally maximized is per capita growth rate. Well, that fits nicely with people's intuitive notion. It fits nicely with Wright's intuitive notion of adaptation. It's like, good. Darwin talked about this notion of adaptation, and people have this idea that selection should cause things to become better over time in some way. Well, here's a precise way in which they're becoming better. You're evolving to a state where the population is maximally able to maintain itself and increase in its environment. So that's why that really appealed to Wright on it. The gradient dynamics also appealed to him, and you made a mention of potential functions. Wright and Fisher, R.A. Fisher, who was also developing this theory around the same time, were very aware of those kind of results. Like for Wright, the importance of potential functions in physics. And so he saw this and was immediately, okay, here's a potential function like dynamic. That grabbed his attention. Now, incidentally, Wright also was able to formally show that if fitness is frequency, if selection is frequency dependent, then this does not necessarily happen. And he didn't really pursue that except to show that it doesn't necessarily happen. And what he did then was say, well, maybe if mean fitness is not being maximized, maybe something else is being maximized. And so he defined what that would be. He called it a fitness function. And he said, okay, we can prove this is maximized if it exists. But it's an integral function and you can't prove that it always exists. So he kind of was left ambiguous about whether anything is necessarily maximized. He kind of wanted to find something that was, but he was careful enough to know he couldn't say that in general, but for frequency independent selection he could. Okay. Now, this is also just sort of a useful way of thinking about, again, frequency independent selection on genotypes when you have something like this. So this example that I gave here, where I drew this with an internal peak, this is what you would get when heterozygotes have higher fitness than either homozygote. Okay? So this might result from a case where, say, this case I'm talking about here, W11 is, I'll scale it 0.5, W12 is 1, W22 is, say, 0.3, something like that. That would produce a function that looks very much like this. How did I eyeball this and pick those out? To make it right, I'd actually have to make this a bit higher. But at this point, when everyone's A1, A1, when P is 1, everyone's A1, A1, then mean population fitness is their fitness. When everyone is A2, A2, then mean population fitness is the fitness of A2, A2. So on one side, it's a little bit higher than the other side. So I made that 0.5 and 0.3. And the fitness of heterozygotes is higher than either of those. Here's the thing that comes out. This always comes up when I teach this to undergraduates in, in introductory genetics or in an introductory evolution course. People always ask, okay, if I set this up, this is not equal to 1. That peak is not at 1, even though I set W12 equal to 1. Why doesn't that go to 1? Just geneticists in the room.
I don't, no. Maybe if you're assuming the random union of gametes and you have everybody heterozygote, in the next generation you will always have homozygotes being produced.
Exactly. So when the allele frequency is intermediate, it doesn't mean everyone's a heterozygote, right? It would be 1 if everybody was A1, A2. Under Hardy-Weinberg conditions, that can never happen for zygotes, right? And in fact, if you force that in a population, so I actually got rid of every one of these homozygotes, like you said, in the very next generation, you're generating a bunch of homozygotes. Half of the offspring produced by those heterozygotes are going to be homozygotes. So in other words, at the boundaries, here, everyone's A2, A2. Here, everyone's A1, A1. In here, there's no place where everyone is A1, A2. Okay? And again, I pointed out because if you find yourself in a situation explaining this to somebody, question's going to come up. It's like, wait, why doesn't it go up to 1? Right? Because I could use those numbers to get that boundary, but not in here. That's the reason. So one thing to mention about this case, the other day when we were talking about inferring selection, someone asked about what about balancing selection. This is balancing selection. Okay? Balancing selection tends to maintain both alleles in the population. When either one gets rare, so if you go down here where A1's getting really rare, you get pushed back. If you get up here where A2 is getting rare, you get pushed back. Okay? And you can use this approach to study all sorts of different cases that you might want to look at. One thing I want to note, I want to consider one other case that is important, because we'll come up with it in a bit. What if we have selection against a recessive allele? Selection against a recessive allele. Remember, a recessive allele means it's expressed only when it's homozygous. In a heterozygote, it's not expressed. So if I say, I'm going to use P for A1, for the frequency of A1. So if I say W11 is, say, 0.2, W12 is 1, W22 is 1. I'll be clear and make that 0.2. This is selection against the recessive allele, right? We're selecting against the A1 allele, but only when it's homozygous, right? So when it's in a heterozygote, it's just fine. That's the meaning of saying it's recessive. Okay. What does this W bar function look like in that case? Well, it's easy to visualize, right? P 0 to 1, the W bar over here. It's easy to visualize when you realize that when P is 0, everyone's A2, A2. So mean population fitness is the fitness of A2, A2, which is 1. So put 1 up here. There you are. When P is 1, everyone's A1, A1. So the mean population fitness is just the fitness of A1, A1, which is 0.2. Might be down around here. And in between them, and I erased that equation for W bar, but you recall it was the Hardy-Weinberg frequencies times these values, which we're taking to not be functions of p. That's a quadratic function. It's got p squared, it has no higher powers of p in it, and it's a polynomial, right? So it's a quadratic. So what shape is it going to be? It's going to be a parabola, a paraboloid shape. And you can kind of guess what goes on here, because the worst possible case for W bar is if everyone is A1A1, right? Because any heterozygotes have much higher fitness. That's going to raise mean population fitness. And so we've got a parabola that starts here, never goes below that value, and ultimately goes up. So what we have is something that kind of looks like this. That's what we would see for selection against a recessive allele. I picked that example for a reason, because we'll discuss that in a moment when we bring mutation into the picture. Does that make sense, this idea? So the key from this discussion here really is that with frequency independent selection, locally you're maximizing mean population fitness. I keep saying locally, that's important. In fact, in this case, there's a global maximum, and in this case, there's a global maximum, and those are the only maxima. But what's it going to look like if heterozygotes have lower fitness than either homozygote? Yes?
I got a little bit confused. In this case, the W12 fitness is always 1, also 1. Does that is that represented in the graph?
Which
the heterozygote one.
The heterozygote has a
Yes.
fitness 1. Yeah. So that, yes, that goes into determining the shape of this curve. In terms of visualizing what the curve is, there's not a point I can put on here that I can immediately infer from that. However, here's what I can infer. Because this heterozygote has much higher fitness than this homozygote, if I start out at this boundary, everyone's A1A1, and I move away from it a little bit, what happens? A few heterozygotes appear. They have much higher fitness than does the homozygote, and so it starts to push it up. Yes?
But if the heterozygotes have 1, W equals 1, shouldn't be like at P equals 1/2 at 1?
Yes, it should. What you're getting at, thank you for asking. I'm standing up here drawing this, and I'm sort of looking at it here at an angle. I drew this wrong, okay? I didn't notice it until you asked your question, and then you started to ask a question. It's like, I see where you're going with this. I drew this wrong. Thank you. Always assume somebody standing at a board is likely to write things or draw things wrong. Okay, I'm looking at everything from an angle, and I'm looking at and sort of drawing over my shoulder. In fact, again, those boundaries are there. This should look like this. So when I say something that seems like, that just sounds backwards, don't think, well, I'm sure he knows what he's talking about, and I'll just write it down. Say, that sounds backwards. And much of the time, it will be. So in fact, the way to think about this, now, the peak is at 1. Okay, so there's not an internal peak here. And the reason to see it is, as I start at this point, if I move in a little bit, I'm creating these heterozygotes. They have much higher fitness, right? So that's pushing it up a lot. By contrast, if I start at this side and I move in a little bit, I'm going from a case where everyone's A2, to a case where there are some heterozygotes, where heterozygotes have the same fitness as A2. I have very few A1 homozygotes initially. And so initially, there's only a very slight drop in W bar until I come out here. Thank you for asking and pointing that out. Is that clear? Okay, apologies for drawing that backwards initially. Now, what this is getting at, again, is that under this condition, frequency independent selection, there are local maxima of W bar, and those local maxima are local equilibriums. And the only other case that I was going to illustrate for you, and you can sort of guess, I'll just leave it as something for you to work out. Come up with a set of genotypic fitnesses that will produce a function that looks like that, okay? And I'll simply leave it at that, but you can sit down, you can probably right now see what would do it, but that's an unofficial homework assignment. You don't have to hand it in. I'm not gonna ask you anything on it, but if it's not immediately obvious right now how to choose values of genotypic fitnesses to get a function that looks like that, sit down and work out how to do it. Okay, and that'll also drive home that functions like this can exist. And in these cases, there are two local equilibria. So where you end up depends on where you start. Okay, and that can easily happen in a case like this. In this case, if I started out here, say at this point, I'm evolving that way. So you end up at p equals 0, which is a local maximum. It is not the global maximum. So this right away tells you that selection, frequency independent selection, does not globally maximize W bar, but it at least locally maximizes W bar. Okay? And that was something that to Wright was very important. And then he came up with all sorts of scenarios about how populations could get across a valley like this. Basically, it required drift to get across a low point like this. That's not really a big area of research anymore because it's easily worked out, but you should be aware of this fact that you can get a case with two local equilibria. So, like I said, Wright demonstrated this, okay, with frequency independent selection, W bar is at least locally maximized. What about with frequency dependent selection? In that case, none of this reasoning works because we got this equation with a dW bar dP here only by assuming that fitness was frequency independent. Those W's that I had here were not functions of P. What if they are? And actually, I'm just going to rewrite that equation over here so I have it up here and I can refer back to it. So I'm just rewriting what I had over there. P times 1 minus P, W1 star minus W2 star over W bar. This equation applies to frequency dependent or independent selection, right? We derived it from the price equation simply by assuming no mutation. So the only assumption beyond no immigration from other populations. So we're dealing with a closed population. The only assumption that we imposed on the price equation to get this was no mutation. Okay? So this should apply to the case of frequency dependent selection as well. Like I said, in the case of frequency-dependent selection, what you should do, or let me rephrase that, your fitness is a function of the frequency of different strategies in the population. Okay? And to illustrate, I just want to do one example to illustrate why frequency-dependent selection does not necessarily maximize W bar. Okay? And so here's a possible scenario. Imagine we have, at a locus, there's an allele, and I'll call it A1, that by itself tends to increase the fitness of individuals that have it relative to others. So it makes them better competitors. But it produces some waste product that builds up in the environment and reduces everyone's fitness. So I'll say allele A1 confers competitive ability, but it produces a waste product that builds up in the environment and reduces everyone's fitness. Produces a waste product that reduces everyone's fitness. So I'm making up sort of a contrived case here, but to illustrate a simple case where we can see frequency-dependent selection not maximizing W bar. And so how might this look? Well, in this case, I might have genotypic fitnesses like this. Let's say W11 is 1.5 minus P. Why did I subtract P? Well, because as A1, P is the frequency of A1, as it increases in frequency, there's more and more of this waste product that reduces everyone's fitness, right? And let's say W12 is 1.3 minus P, and W22 is 1.1 minus P. So in other words, if we ignore the effect of this waste product, ignore that, there's linear selection for the A1 allele. W11 is higher than W12, higher than W22. But everyone's fitness is reduced as the A1 allele increases in frequency because it's doing something that's harming the environment, that's in the environment that's harming everybody in it. So this would be an example of frequency-dependent selection. And just to drive home, what that means, I said it, but I didn't write it before, that these genotypic fitnesses are functions of P, right? And you can see if you plug these into the W bar equation and took the derivative of it, you wouldn't get the same thing you got before because you'd have to differentiate these with respect to P as well. Before you didn't have to. So let's consider that case. Well, this equation should still apply to it. Okay? And the key, what matters, is the difference between these marginal fitnesses. So P times 1 minus P is non-negative, W bar is non-negative. So what matters for the direction of evolution is this difference between these two. So what do we come out with for that? You can write out an equation for W1 star, and it still is going to be P W11 plus 1 minus P W12. Okay? And I'll let you confirm from that equation and from those that we end up with W1 star comes out to be 1.3 minus 0.8 P. And W2 star, that's W1 star, W2 star comes out to be 1.1 minus 0.8 P. Notice the slope with respect to P is the same. Let's plot it. So now I'm going to plot both of those as a function of P. P goes from 0 to 1. Here I'm putting W star, marginal fitnesses, so I'll have two lines for them. Okay? For the A1 allele, W1 star, it starts out at 1.3, so if I put 1 about here, it starts out about up here. And it declines when P is 1, is 1.3 minus 0.8, which is about 0.5, is 0.5. So that would be about over here. So this should be a straight line. And again, my skewed drawing skills fail to make it a straight line, but you know it's a straight line that looks like that. That is W1 star. What about W2 star? Well, it starts out at 1.1, so it starts out about here. And it drops off with exactly the same slope. So it's dropping off like that. What's going on in this case? Well, remember, change in allele frequency is determined, the direction is determined by W1 star minus W2 star. So as long as W1 star is greater than W2 star, the A1 allele will be increasing in frequency. Well, it's greater everywhere here. This is W2 star. Here, this line is above this line everywhere, right? So you're going to evolve this way. Note, by the way, I drew short lines at the end and a long line in the middle. Why did I do that? What in this equation gives you that? Right, I drew this line much longer than that line and that line. What about this equation tells you that they are?
The peak of the variation is at probably equal to 1/2 in the first term. There.
So you said the variation, you mean that term, p times 1 minus p. That is the variance. And yes, that has a peak at 1/2. And that goes to 0 at the boundaries, right? So this term is what's determining the length of these vectors. Because the difference, W1 star minus W2 star, in this case is a constant, okay? And this is the reason I'm bringing it up is, first of all, to make you look back at that equation and think about it, but also to start to point out at the boundaries, near 0 and 1, p times 1 minus p gets very small. And as a consequence, selection gets relatively weak at the boundaries. So you keep evolving this way. The A1 allele increases in frequency. What is mean population fitness in a case like this? Well, because these are straight lines, right? It's the mean of the two. It's the mean weighted by p. It starts out here, right? Because when p is 0, everyone's A2, A2. So mean population fitness is just W2 star, right? When p is 1, everyone's A1, A1. So mean population fitness is going to be W1 star. And because I made up a case where everything's linear, this is a straight line. It goes like that. So what happens over time, due to selection, is the A1 allele increases in frequency and mean population fitness goes down. Furthermore, note, after this has happened for these numbers I picked, everyone's fitness is lower than anyone's fitness when we started out. So selection is not only reducing W bar, it's not only reducing mean population fitness, it's actually causing the entire population to become less fit, in a sense. And there's no reason that this could not reduce W bar, mean population fitness, to the point that the population starts declining and goes extinct. So in fact, it's perfectly plausible that natural selection, favoring those things that at the moment do better than anything else in their environment at surviving and reproducing, will lead the population to evolve to a state where it goes extinct. And there aren't a lot of good examples that we know of in nature of this happening, but people studied the potential of this to happen, and it's sometimes called Darwinian extinction. The idea being that it's Darwinian selection, but it's leading the population to a point where the entire population goes extinct. So, what this example is sufficient to show is that with frequency-dependent selection, W bar does not have to be maximized. And hopefully this gives you a sort of an intuitive idea of a case where it wouldn't be, right? And it also drives home why. Because what matters is the difference between these, not the absolute values of them, but the difference between them. As long as a strategy consistently does better than other strategies, it increases, even if it increasing is causing its and everybody else's fitness to go down. That's perfectly plausible. Does that make sense as the case? So this is sufficient to illustrate that frequency-dependent selection doesn't necessarily maximize W bar. But does it maximize something? Wright had this hunch that it probably did, but he couldn't prove that it did. And a lot of people have had that hunch. In fact, probably the single most common misconception about evolution by selection, and I'm giving a hint as to what the answer to this is going to be, that it's a misconception, but it's a very common misconception, is that somehow something is constantly getting better. There's some quantity that is being increased over time by selection, or put another way, things are always evolving to get, in some sense, better. People don't know what better means. One notion of better might be higher W bar. That would make intuitive sense. Okay, we know that's not that, but maybe there's something else, right? In this example, there is an equilibrium, right? There's a stable equilibrium in this case, and that means there's a Lyapunov function, if you're into dynamical systems theory, for this, and so you can construct a function that has a maximum at that point, right? In fact, I could write any number of functions easily that have a maximum value at that point. So can I prove that there is not some function that in the process of it being maximized is driving evolution there? In fact, in evolutionary game theory that we're not really talking about much here, but equations that look just like this come up in evolutionary game theory all the time. You know what the big difference is? They don't have that variance term, right? In game theory, you just look at the difference between these two. So you're not concerned in evolutionary game theory with the rate at which you're going anywhere, but you're concerned with where the equilibria are. One approach to evolutionary game theory is to come up with a fitness, they call it a G function or a fitness generating function, that is maximized at equilibria, right? So you could construct a function that is maximized at this point that's based on these fitness values, these fitness functions, and say, okay, it's not W bar that's being maximized, but that is. And it means that even if the population is evolving to lower fitness, in some sense, that strategy does better than other strategies, and so it's being fixed. So game theory, too, gives hope to the idea that universally there might be some function that is always maximized. There's not. How do we know there's not? Well, this gets back to the dynamics question that came up earlier. You can construct, with frequency dependence, I can make, basically, these have any function of p that I want, okay? So in a sense, this is one of these cases where people divide things into like two categories. People like dividing things into categories like vertebrates and invertebrates, right? That always bothered me because I was an invertebrate zoologist. And of course, invertebrates just means all animals that are not vertebrates, okay? And that's this massively diverse group of organisms. And they are not a phylogenetically distinct group. They're highly paraphyletic. In fact, there's this huge tree of metazoans, of animals, and vertebrates are one little branch, one little twig on that tree. But people divide it up into vertebrates and invertebrates, sort of like those are equal. The same thing goes here. Frequency-independent selection is an incredibly narrow special case. That's where these values are constants. Frequency-dependent selection includes every imaginable function of p that I could put in here. So that's the big case, right? Almost all systems, just if you pick things at random, would be like that. And so I made up a really simple case here. But you can make up more complicated ones. I'm going to give you one. To discuss this, I'm going to just write fitnesses in terms of the frequencies of genotypes. And just to make it easier to write, I'm going to give them names. So I'll say gamma 11 is the frequency of A1, A1. Gamma 1, 2, similarly, frequency of A1, A2, and gamma 2, 2, frequency A2, A2. So I don't have to write out all the 2p times 1 minus p's and things like that in there. Also, of course, this allows me to generalize beyond the assumptions of Hardy-Weinberg, because I'm no longer demanding that genotypes be in Hardy-Weinberg frequencies. And then let's make up, and this is a bit contrived, I made it up to illustrate the point, but you'll see why that's sufficient to illustrate the point. Let's consider these fitness functions. W11 is 1 minus 3 gamma 1, 2 plus 3 gamma 2, 2. W12 is 1 minus S. S is going to be a variable that I'll tune. S times gamma 1, 2. And W22 is 1 minus 3 gamma 1, 2 plus 3 gamma 1, 1. So I made this up basically to be symmetrical. And the idea here is heterozygotes reduce everybody else's fitness. So heterozygotes are doing something that reduces everyone's fitness, including the fitness of homozygotes and including their own fitness. How much they reduce their own fitness, I made that a variable that I can tune. And the homozygotes each benefit from one another. So if you're W11, it's good to have or if you're A1A1, it's good to have A2A2s around, right? If you're A2A2, it's good to have A1A1s around. So the two homozygotes benefit one another, but heterozygotes reduce everyone's fitness, okay? In that case, depending on what I pick for S, I can get all sorts of kinds of dynamics. I'll give one example. And here, as before, in answering the question from up here, I'm going to plot the frequency of A1 at time t and the frequency of A1 at time t plus 1. These both go from 0 to 1. Y'all have done ODEs, ordinary differential equations, and some dynamic stuff. This is a time one map. Is it obvious why I'm drawing this? Have you in this class, have you seen time one maps like this? You have? Okay, good. So then you'll know why I'm going to draw 45-degree line like that. And it's skewed somewhat, but see, when I look at it from this angle, it looks square. You know the painting by, was it Hans Holbein, the younger, the ambassadors? He was the court painter to Henry VIII, but he did this great painting, and it's really cool. You should look at it, called The Ambassadors. It's of these two guys. One was an ambassador from the Vatican. The other was a cleric in England. And they were having these secret meetings because Henry VIII was trying to separate from the Catholic Church. But anyway, those guys were meeting, and one of them commissioned Holbein to draw a portrait of them. And so it has a portrait of them looking like these, dignified gentlemen. And they've got all these things around them that show that they are highly learned, right? There are all these scientific implements and musical instruments and things showing how sophisticated they are. And then floating in front of them is this deformed skull that's floating in the air. And you can tell that it's a skull, but it's really stretched out and deformed. And of course, the symbolism is that even for these highly sophisticated, learned gentlemen, death is ever present, right? It's always floating in the air around them. And if you get to where that painting is, or if you even see a good reproduction of it, turn the angle, and it's really quite impressive. When you look at it from the right angle, the skull looks not distorted at all. It looks just perfect. And I didn't realize that. I had seen it and thought, okay, there's some symbolism of being distorted. No, it's when you look at it from the right angle, it looks perfect. one of the things that I like about it is, and he was the court painter to Henry VIII, right? This is basically surrealism. And they liked it. One of those two guys, I don't remember which one, but one of those two guys commissioned it, and he really liked it. And he put it in his house, and he passed it on, and it was like, hey, check it out. Hans Holbein painted me. And illustrated that death is ever present. Yes, I'm going to die, like everybody. Right? Holbein said so. So anyway, that's a good illustration. When you look at something from the different angles, things get distorted. So what function do we get for a time one map for these? Well, if I take s equal to 2, I basically wind up with a function that looks like that. Actually, it goes like this. And the slope of this line, where it crosses that yellow line, that identity line, is steeper than -1. So this point is not stable, right? You bounce around out from that. This is a case where there is a stable limit cycle, and it kind of looks like this, roughly. So s equals 2 yields a stable limit cycle. Now, if I make s bigger, if I tune it up more, what happens is this gets steeper and steeper, and you go through period doubling, and you can get chaotic dynamics out of it. But that's not the interesting case. The really compelling case is the stable limit cycle. Because think about it, what does that mean? I'm going to draw it like as a time series, time p over time, 0 to 1. It means that initially, I might have some transients, but then I settle into something that looks like this. And this is a discrete time system, so in fact, you're jumping back and forth between these points. I'm drawing the lines just to illustrate the connections between the points. So the system is jumping back and forth between those points. Okay. What if, so is it possible in this case that there is some gradient dynamics, that there's some function with the property that every generation increasing that function is what's driving evolution? Now keep in mind, it has to be an autonomous function. It can't be a function of time, right? Because of course, time is always going on. But rather, it has to be some function of these fitness functions without time in it. So it can be a function of p's, right? But not time, and a function of whatever I choose for this. Well, there really can't, right? Because if there was, it would mean when I start out here, what makes me go here up to this point must be that that function is increasing, because increase in that function is what's driving evolution. Fine. But the next generation, I come back to where I started, which means increasing that function would have to take me back where I started. But you can't do that, because that function must have the same value here and here. So it can't keep increasing and take you back and forth like this. No gradient function like that, no potential function, no fitness generating function in evolutionary game theory could generate this dynamic of a stable limit cycle. And remember I said, you can make this chaotic if you make s bigger. And when I was first playing with this, that seemed like the cool thing. Chaos is cool, right? So it's like, oh, looking at chaotic dynamics. That's actually less compelling, because a chaotic system never repeats. So you could construct a set. It would be a fractal set. It wouldn't be continuous. But you could construct a set on 0 to 1 that would have the property, at least from a given starting point, for a chaotic system, that set function of these things is always increasing, because it never repeats itself. But a limit cycle does exactly repeat itself. It comes back to exactly the same point it started at. And that confirms that there could be no function that has the property that evolution is driven by that function increasing. Does that make sense? And I emphasize this because you might think this seems like a very sort of abstract thing to even make a big point of. Why am I harping on it? Because like I said, a lot of people, including Sewell Wright, including most of the evolutionary game theory folks, and including most biologists and scientists of other sorts and people in general thinking about evolution, their intuition is behind it all, there's something getting better. There's something that's increasing that's driving evolution. Okay? A single example of a system like this that has a stable limit cycle is sufficient to prove that in general that is not true. There is nothing that is universally increased in evolution by selection. Sometimes some particular value might be. In some special cases, W bar is, right? In other cases, like this, W bar is not, but there exists some G function, some fitness generating function that does increase, and it's a function of these, and it increases. And then the temptation is to think, okay, can I come up with a general description that would unify that with the W bar case? No, you can't, because in fact there exist cases where there is no such function. So do not think of evolution as a process that involves anything necessarily getting better, and do not think of it as a process that's driven by there being any function that's always increasing. Evolution does have laws. The price equation I gave you the other day is a law. Right? That's true in all evolving systems everywhere, exactly. But the laws do not involve some function or some quantity that's always increasing. Does that make sense? And I use this example also to illustrate, this is a question that has come to the minds of a lot of biologists. Is there something like that? Many of them assume that there probably is, but they didn't know what it was. No amount of experimentation, no empirical observation in nature, no experiment in the lab could possibly confirm that there is no such function. This example confirms that there is no such function. So this is a case where something that seemed intuitive to a lot of biologists turns out not to be the case, but the only way to demonstrate that, the only way to prove it, is with a theoretical analysis looking at a critical case. Does that make sense? Does that seem compelling? Okay. If you want to play with these equations, you're welcome to. Like I say, as you make S bigger, you can get chaos. You can get period doubling, so it looks like classic period doubling to chaos. And because it's symmetrical, it looks nice and neat. As you make S smaller, what happens is this starts to flatten out. And in response to the earlier question about the frequency independent case, with frequency independent selection, you can never get this to go past a point like that. You can never get this to cross with a negative slope. And so that equilibrium is always going to be stable. Yeah.
I'm sorry, I understood the conclusions and the equations, but it isn't still clear for me. How do you get from the equations to that conclusion?
Right, so from the equations, we get the fact that there's a limit cycle. Okay, that's the key. So I'm not going from the equations to the conclusion, I'm going from the existence of a limit cycle to the conclusion. And the conclusion is that there is no function that has the property that increasing that function is what's driving evolution, right? It would have to be a function of these. It has to be an autonomous function, not a function of time, okay? So it has to be a function of these. And if it existed, it would have to be the case that when you're at this point, it has some value, and then it goes up to get you to this point. And then it goes up again to get you to this point. And it goes up again to get you to this point, and so forth. However, this point and this point are the same. So they have the same value of p. And so for any function that's just a function of p, they must have the same value. So you couldn't increase here and increase here for a function that's just a function of p, because it would have to come back to, it has to come back to where it started. Does that make sense? That's the idea. So yeah, I was not drawing the conclusion directly from those. I'm saying those give you a limit cycle, and a limit cycle gives you the conclusion. Yeah.
I have a question about, in this case, we were working with a continuous model, and in this case, we analyze a discrete model. So how did we achieve the discrete model?
Okay, this was a discrete model as well.
As well?
Yes.
Okay.
Yes. So this is going over some time intervals treated as one generation. So that's why I'm writing this not as a derivative of p, but as capital delta p, the change over that time interval.
Okay.
So all of these are discrete.
Okay, and the time step is the generation.
Yes.
Okay,
Yeah, all these are discrete.
thank you.
This is a good point. And so you can, and we're not going to go into it, it doesn't really yield more insight, but you can write a continuous time version of this perfectly well. If I made a continuous time version of this univariate equation, it's just a function of p, right? And now these w's might be functions of p, that's fine, but it is a function of p. Could I get a limit cycle out of it? For a one-dimensional dynamical system. Could not get a limit cycle in that case, right? I would have to make it two-dimensional. If I made it two-dimensional, I could. So for instance, if I introduced, to do that, I would have to introduce another locus. And I've got a allele at this locus and an allele at this locus, and then I can make a two-dimensional limit cycle. But I stuck with discrete because it's easier simply to write out the case for a one-dimensional case. Yeah?
Could we have chaos?
Yes? In two dimensions, oh yeah. Well, actually, no. So continuous in two dimensions, no, it would not be chaotic. But you can get a cycle from it. 3 dimensions for continuous, then you could get chaos. Does that make sense? Just general, these are general properties of dynamical systems. So one-dimensional continuous, what would that require? Okay, we're stepping away a bit from just this topic, but this kind of dynamics comes up everywhere. So if I had one-dimensional continuous, what would it require to get a limit cycle? My state space is a line, right? So a continuous limit cycle would have to go back and forth on a line, but visiting every point in between there. That would require, sometimes at a particular point, the derivative is pushing you that way, sometimes it's pushing you that way. For an equation that is autonomous in a sense, so it's truly one-dimensional, that's impossible for a continuous case. For a discrete case, it's possible because you can simply jump like that, okay? In 2 dimensions, though, continuous, I can get a cycle just fine. However, in 2 dimensions, could I do something that did this? No, because at that point, if I only have 2 dimensions to deal with, it would mean sometimes I'm going that way and sometimes I'm going that way at that point. And so I could not get a cycle higher than that 2 cycle, right? So you couldn't go on to chaos. How would I do that? Add a 3rd dimension. Add a 3rd dimension, I can go anywhere and miss myself, and the line never has to cross itself. So for a continuous system, you don't see limit cycles in 1 dimension, or you don't see limit cycles in 1 dimension. You can see them in 2 dimensions. You don't see chaos until you go to 3 dimensions for a truly continuous system, unless you add some other thing, but essentially that's making it higher dimensional. So the classic thing in biology that comes up that you would add is a time delay, right? 2 dimensions with time delay, you can get chaos. But the reason then is essentially what matters is not just where you are now, it's where you were at some time in the past. So if I'm coming this way to that point, I'm using not just the fact that I'm there, but that last week I was here. In a sense, that's adding another dimension, right? So if it's truly two-dimensional, you can't get chaos, but you can get a limit cycle. Make sense? Okay. So, questions? Okay. So this is just getting at things that we can infer. These are really simple models. Even though we started out with the price equation, when we're putting in these assumptions like random mating and especially frequency independent selection, that imposes a major simplification. But we can get results that tell us fundamental things about the process of evolution. Frequency independent selection can locally maximize W bar. Frequency dependent selection in general does not. And in the most general case, there's nothing that is necessarily maximized. What I want to do now is extend this a bit. Remember the main assumption that went into this equation did not require Hardy-Weinberg frequencies, so it does not require random mating, okay? But we left mutation out of it completely. And just to go back to the price equation, change in mean phenotype, okay? We can write that as 1 over mean population fitness times the covariance between phenotype and fitness plus, remember I had that lowercase delta, the covariance between difference between offspring and parents and fitness. And then I have to do plus delta bar, the mean difference between offspring and parents. And remember, this lowercase delta was offspring phenotype minus parent phenotype for an individual. So it's property of an individual. It's basically just writing your offspring phenotype as yours plus a difference. And I said that this term is capturing just the effects of differential survival and reproduction. The terms that have delta in them are capturing other things that are happening during reproduction. This one in here is interesting. It's almost always ignored. We'll see tomorrow. There are cases where you shouldn't ignore it. But for the moment, I'll point out this is the covariance across the population between difference between your offspring and your fitness. And that means this will be non-zero only when the difference between your offspring and you is a function of how many offspring you produce. Okay, that can happen sometimes. But in general population genetic circumstances, it's assumed that that's not happening. However, mutation overall is happening. Now, mutation is generally not going to enter into here because mutation happens regardless of how many offspring you're producing, right? So it's not going to be a function of W. But if mutation's happening, we need to include it in this term here. So I want to address how mutation comes into here. And the example that I'll give, very simple case, let's still consider two alleles. So I have allele A1 and allele A2, and I'm now going to allow them to mutate back and forth. A2 can mutate to A1 at a rate mu sub 1, and A1 can mutate to A2 at a rate mu sub 2. I'm choosing subscripts to be the allele that you're going to. So mu sub 1 is the rate at which A1 is appearing due to mutation, and mu sub 2 is the rate at which A2 is appearing due to mutation. Okay, so this is a very simple model of mutation. In that case, what would this delta bar look like? Well, it is the average across the whole population of the change in phenotype, but remember now our phenotype is whether or not you're A1, right? We set that up yesterday. due to mutation. And you're going to stop being A1 due to mutation away from it, and A1s are going to appear due to mutation to it. At what rate do they appear? Well, the frequency of A2, 1 minus P, times mu1. That's the rate at which new A1s are appearing due to mutation. Notice I had to multiply by the frequency of A2s. If there were no A2s in the population, if you're fixed at A1, no new A1s can appear, right? So the rate at which they're appearing has to include not just the mutation rate if there were A2s, you have to multiply by the frequency of A2. So this is the frequency of A2 times the rate at which it mutates to A1, but you're also losing A1s at a rate P mu2. The frequency of A1 times the rate at which they're mutating away to A2. So that's our delta bar. And so with that in mind, I'm going to stick with this version that we derived from the price equation by saying that phenotype is whether you're A1 or A2 in this case. Let's now write delta P is P times 1 minus P, W1 star minus W2 star over W bar, plus, it would be plus that delta bar, but it's that. So plus 1 minus P mu1 minus P mu2. That's the change, including both selection and mutation. Now, when people are studying this, in general, mutation is taken to be a very weak process, right? So in a given generation, especially if you're in some intermediate state in there where P is intermediate between 0 and 1, mutation is generally assumed to be very weak. The interesting question, though, is what happens when an allele that's being selected against is really rare. And here, I want to go back to that diagram that I drew wrong earlier. 0 to 1, P, W bar. So even we're considering now frequency independent selection. When we had selection against a recessive allele, we ended up with a function that look like that. And the rate of evolution, if we ignore mutation, just considering selection, is a function of the slope of this function and P times 1 minus P. Well, when I get down here close to 0, that slope is really low. And P times 1 minus P is really low. And so, in other words, this is a case where selection's pushing you away, is pushing you ultimately towards P equals 0, but it's getting incredibly weak down here. Weak enough that mutation might be able to have an effect here. Because, think about that equation that I gave for mutation here. This function, I'll draw it like this. Here's P from 0 to 1, and I'll write delta bar here. When P is 0, this term is 0, and this is just mu1, so it's some positive mu1. When P is, there's a 1, when P is 1, then this term is 0, and so it's minus mu2. So it's minus mu2, which is something down here. And it's a linear function, so it goes like that. In other words, it is maximally, the effects of mutation are maximized at the boundaries, at 0 or 1. At 0, when P is 0, then you're creating A1s at the highest possible rate. When P is 1, so everyone's A1, then you're eliminating him at the highest possible rate. So whereas selection gets weak at the boundaries, mutation gets strong, is strongest at the boundaries. And this leads us to want to combine selection and mutation into the same equation. And the way to do it, the way people approach this, is to basically take this equation and solve for the equilibrium. So you solve for mutation, selection, equilibrium. Okay. And so let's consider a case that might give this. There would be of interest in this case. Let's consider the case that we talked about, selection against a recessive. So I'm going to say that W11, now this is the A1 allele. I'm selecting against the A1 allele, right? W11 is the fitness is, I'll call it 1 minus S, where S will be a variable that we can tune. And W12, because A1's recessive, is going to be 1. W22 is going to be 1. Yes?
Mu1 and mu2 are rates, or?
Yes.
Okay. For each allele.
Yes. They're the overall rate at which, yeah, so you think of it as a probability that an A1 will mutate to an A2, or an A2 will mutate to an A1. They're referred to as mutation rates in that sense. Okay, so set up those cases, right? And then from those, we can solve for the marginal fitnesses. And I'll put this up and you can double check me. W1 star, in this case, comes out to be 1 minus SP. And W2 star comes out to be 1. And it should make sense that W2 star is 1 because all the genotypes that have A2 in them have a fitness of 1. So its marginal fitness is just the average over those, but they all have the same fitness. So it's an easy case. It has a fitness of 1. Okay. And if we work out what W bar is in this case, mean population fitness, because we have that in the denominator there, that comes out to be 1 minus SP squared. Now, we're interested in cases where P is rare. P, or where P is low, so A1 is rare. A1 is being selected against in this case. Okay? So we expect it to have a low frequency. Mutation may be pushing it back a bit, but it's being selected against. So we're going to be interested in cases where P is quite small. Now we clip into population genetics mode, which very happily says, oh, well, P is a very small number. Squared is really small, and so we can ignore it. So we can say W bar is approximately equal to 1. Okay? That's only valid when you're talking about very low frequencies of the A1 allele, but we will be considering only very low frequencies of the A1 allele. Okay? In this case, also, W1 star is this, W2 star is this, so putting those together, W1 star minus W2 star is equal to simply minus SP. And so we end up with delta P is approximately equal to P times 1 minus P times minus SP plus 1 minus P mu1 minus P mu2. And again, population genetics mode, you say, okay, P is a small number. We already said that. P squared is very small. Mu is a really tiny number. So I'm multiplying two very small numbers together. Here, because P is small, 1 minus P is close to 1. right? But here I'm multiplying two very tiny numbers together, so we'll ignore that. This is the population genetics approach. And we basically have these parts. And we want to find a case where delta P is 0. We want to find an equilibrium. Now, strictly, we'd have to test for the stability of that equilibrium separately. We'll do that later, or you can check on it for yourself. It turns out this condition holds. Notice this is a minus sign here. This implies 1 minus P mu 1 is equal to SP squared times 1 minus P at equilibrium. And because I'm setting it at equilibrium, I'm going to put a hat over these P's to indicate that this is the equilibrium value of P. Given that, you can solve for that P hat, and it comes out to be the square root of mu 1 over S. And that then is our equation for the mutation selection equilibrium when we have selection against a recessive allele. Does that make sense how we got that? I kind of went quick through this. We'll pick up and talk about the meaning of this at the start of discussion tomorrow. Before then, if you're looking at this saying, okay, I'll take your word for it that W1 is 1 minus SP, sit down and confirm it. Okay, so if you just check the steps in here, then it'll be fresh in your mind, and tomorrow at the start, we'll come back and talk about the biological significance of that equation. But it's 10:48, so I'll stop there. And we can tell the coffee people if they're out here that they can come down. You can come. It's okay with me if you set up while I'm talking, and that remind me that I'm running out of time. So, what? No, I'm saying at first she came in when I was lecturing and set up, and that was okay. And then I've